life, I wish you well. I wish you peace. I hope that we'll find harmony. I wish you good.
How did it all begin? That's a heavy question. A question that we don't have a complete answer for. That's right, if you clicked on this video thinking I would be telling you exactly how life on Earth originated, then tough nuggets. Hi, I'm Vi the Neutrophil, and this channel is all about exploring the ways in which biology is beautiful and nuanced. And you're not getting a straightforward answer from me, even if you dangled me over a pit of open Legos. Most of my videos thus far have been firmly rooted in experimental molecular biology. It's the lane I feel the most comfortable in. But asking the big questions like how did life on Earth originate is a question that demands an academic tradition rooted in historical scholarship in addition to experimental practice. Think about it for a second. The only way to know with 100% certainty how life on Earth originated is to invent time travel and to know which location or which locations harbored life without having to interact with said system. There are questions from history that share this same epistemological limitation. There's no way to know with 100% certainty what Gilgamesh looked like, some interpretations more muscular than others, but by gathering supporting evidence, we can kind of narrow down the possibilities and assign weights and probabilities. We're just going to have to embrace and accept this level of ambiguity. And also, keep in mind that my training is in the realm of molecular biology. Abiogenesis is an incredibly multidisciplinary subject, including physics, astrobiology, statistics, geology, paleontology, chemistry, biochemistry, molecular biology, ecology, zoology, marine biology, even philosophy. Ugh. Even two molecular biologists are going to have a different perspective here. The goal of this video isn't to give you the origin of life, but to the best of my ability, the most modern, cutting-edge models by which life may have originated. Got it? Then let's begin. Weirdly enough, a video about the start of life turns out to be really difficult to start. I mean, where do you even begin? With the Big Bang? After all, chemical synthesis happening in stars is really the first event that kicks off the whole life thing. But to save you, and well, mostly myself, from the madness that is space, I'm going to do a fair bit of fast forwarding to already having a habitable Earth. From this starting point, we actually have a picture of how life formed. That picture is, of course, rendered on a toaster given how blurry the details are, but logically speaking, there isn't likely a progression that doesn't look something like the following. On a habitable Earth, natural spontaneous synthesis of organic material eventually created the first protocell, a collection of biomolecules encased in a membrane. And from here,
functional polymers.
And our last universal ancestor or ancestors radiated out to what Darwin called endless forms most beautiful and most wonderful that are being evolved. So the rest of this video is going to focus on trying to organize the absolute chaos known as abiogenesis. And to do that, I'm going to split the question into two. What were the chemical prerequisites for life to happen? And once those prerequisites were met, how did evolution then give rise to the first life form? How do we know life began with prebiotic synthesis? If you think about it, it kind of logically makes sense. If you buy into the idea that life radiated out of one or several instances of individual organisms, then it logically follows that before the first instance of life was non-life. Noted smart man Aristotle had conceived of this, noting that animals like flies seem to just spontaneously generate within flesh. And while Aristotle had the spirit, he wasn't exactly right on the money. The idea of life springing from non-living matter wasn't wrong, he just got the particulars a bit mixed up. Louis Pasteur, a man with a gaze so strong it sterilizes microbes, showed that after sterilization and protection that broth would not spontaneously give rise to life. That only when exposed to the environment does broth gain the ability to generate life through what we would call inoculation, or if you're an undergraduate researcher, you call it contamination. So we have a bit of a paradox here. At some point, life had to have sprung from non-life, unless you believe in the whole we got loaded in with console commands hypothesis. But in the laboratory, we have only seen life spawn from life. I've noticed that there are two rough overlapping frameworks of how to conceptualize a valid path from chemistry to life, and that is through breaking the prebiotic chemical synthesis question into two. The first one being location, location, location. Where can we find the satisfactory chemistry required to make bioactive molecules? And next, what or which biomolecules were first synthesized? All of these hypotheses can coexist to some degree, but we will have to judge them all based on some shared criteria. In order for a hypothesis of prebiotic synthesis to work, we need to account for a couple of conditions that define life as we know it. Note, this is a non-exhaustive list because I'd like to finish this video before I apoptose. Each hypothesis has to have an answer for the origin of 1. Water, as it provides the medium in which biochemical catalysis can happen. 2. A carbon source, the molecule that all life is built upon. 3. An accessible nitrogen source, which is a key element in building proteins and the molecules of information, DNA and RNA. And, if you didn't already know, the amino in amino acid refers to a nitrogen-containing molecule. 4. A bioavailable phosphorus source. Phosphate is important for building all sorts of biomolecules, namely the molecules of information, the cellular energy currency ATP, and phospholipids, which make up our modern cell membranes. 5. Speaking of membranes, we need amphiphiles to form primitive cells. Amphiphiles are molecules with a water-loving component and a water-hating component, naturally giving them the property of forming little compartments. I just mentioned a particular type of amphiphile, the lipid. Our modern cells are made with phospholipids, which almost certainly were not a thing back in the early days of life for reasons. We'll get into the origin of phospholipids later, but for now, a reasonable standard to hold these models up to is the generation of the long-chain fatty acid, the most plausible precursor to the modern lipid and functioning amphiphiles on their own. 6. The right chemistry to form heritable molecules of information, which we're going to assume is RNA. We know our modern biology relies on DNA for stable long-term storage, but the chemistry required to make DNA was likely not accessible on the early Earth. DNA is read to make RNA, which is then read to make protein, so RNA, being a molecule that can store information, is our genetic molecule of choice. 7. And finally, some explanation for the synthesis of amino acids, the building blocks of protein. We'll start off by discussing frameworks that prioritize where life originated, starting with the sky. Specifically, we're going to talk about atmospheric chemistry and how the sky could contribute to the primordial soup model. Oparin and Haldane back in the 1920s independently noticed that the reducing atmosphere of the early Earth would have made an excellent chemical reactor to generate simple organic compounds. A reducing atmosphere simply means this environment really wants to create chemical reactions that add electrons onto things. You'll see why this is so important in just a little bit. In 1952, Miller and Urey decided to test whether this reducing atmosphere would have been sufficient to synthesize the building blocks of life. So they made some bottled early Earth atmosphere modeled after the atmosphere of gas giants and sent periodic zaps through the reaction vessel. And what they found will shock you. Over the course of a week, the reaction mixture, which started vaguely pink, darkened over time. And when Miller and Urey analyzed the products of the vessel, they had found glycine, alanine, and aspartic acid, 3 of the 22 modern proteogenic amino acids our proteins are made out of today. Miller and Urey had just made history proving that organics could literally rain from the sky. This sky reactor would drip organics into the ocean, and over time, according to Oparin and Haldane, the ocean would become a hot, dilute soup of organics, and through some mechanism not yet known, eventually give rise to the first life forms. So let's take our checklist and see how this soup performs under scrutiny. Okay, first on the list is water, which is a resounding yes. The ocean is made out of water. And to save you some time, the rest of these hypotheses are going to have water too. Second is carbon, which is also a check. Early Earth was a nightmare hellscape. Volcanic activity and magma oceans would have dumped carbon dioxide and carbon monoxide way up into the atmosphere. Hold up, magma oceans? The early Earth was metal, man. Over time, the Earth would cool, forming the world's first water-based oceans. Atmospheric gaseous CO2 would have been pulled out from the atmosphere and into the oceans, meaning oceans would have had plenty of dissolved carbon dioxide. Third is nitrogen, and nitrogen is kind of a problem. Miller and Urey's experiment revolutionized the field of abiogenesis, but the results hinged on one very important assumption. Miller and Urey were assuming that our early Earth atmosphere was just like those gas giants. Gas giants do have ammonia as a part of their atmosphere, which is really convenient for abiogenesis because ammonia is a bioavailable form of nitrogen. And while that would have been nice for Earth at the time, we now have a better understanding of our ancient atmosphere, and nitrogen was in the air primarily as N2 from volcanic activity. This is a problem because N2 is super stable. The triple bond between diatomic nitrogen is one of the strongest in all of chemistry. Nowadays, plants are reliant on symbiotic bacteria to help them break down this gaseous nitrogen enzymatically into that usable ammonia. This doesn't spell the end for our sky reactor though, as alternate paths to making usable nitrogen species involve bombarding that gaseous nitrogen with UV from the sun to form hydrogen cyanide and nitrogen-oxygen species. Neither of these molecules is as bioavailable as ammonia, but weirdly enough, with some additional chemical magic, both can be converted into useful organics. Cyanide, yes, that cyanide, known for its deadly properties, can undergo some goofy-odd chemistry to form adenine, one of the 5 nucleobases needed for the generation of DNA and RNA. Fourth is phosphorus. Phosphorus, as you're going to see, is kind of a final boss in abiogenesis models, often called the bottleneck element. Phosphorus, as it naturally exists, is bio-unavailable. Mineral phosphate is usually insoluble, meaning it is really good at staying solid when in water. This is a problem because in order for productive chemistry to happen, you need to solubilize phosphorus. You can't form ATP if that phosphorus is clinging onto calcium for dear life. Once life starts happening, the problem of insoluble phosphorus is solved. Enzymes can catalyze the formation of organophosphates. But before that, liberating phosphorus is going to be a serious problem. And it's a problem that the primordial soup doesn't really have a great answer for. Because mineral phosphorus is so hard to liberate and the ocean is very big, accumulating enough phosphorus in a local area to do anything with is going to be near impossible. And even if you did somehow get some liberated phosphorus into the ocean is rich in calcium, which can react with that phosphate, once again forming an insoluble, inaccessible form of phosphorus. A perpetual exercise in futility. Now, there are some workarounds for this problem for the primordial soup, but we'll get to that in the future. Fifth is amphiphiles, protolipids to form the boundaries of our protocells. Here's where atmospheric chemistry alone really struggles. Miller and Urey didn't actually find fatty acids in their bottle of organics. Fatty acids require an acid part and a fatty part, a long chain of carbons that would prefer to bind each other than to bind to water. I couldn't find any instance of a Miller-Urey type experiment generating an organic acid long enough to form something close to our modern-day cells. Sixth is synthesis of an information molecule like RNA. We've already seen that photochemistry in the atmosphere can lead to the production of one of the nucleobases needed to build RNA. Other nucleobases are possible, but to my knowledge, there's no way the sky itself can stitch together these bases to form RNA proper. Seventh is the origin of amino acids, and this one is something we've already covered. Miller and Urey only found 3 of our modern proteogenic amino acids in their soup, but a reanalysis of their experiment expands that list to 6 amino acids. Modified Miller-Urey experiments further expand that list to 10, meaning about half of the protein building blocks modern life uses today had the potential to be pulled from the sky. Pretty compelling stuff for what most people would call the first serious model for abiogenesis. The primordial soup was hugely impactful in the world of abiogenesis. Miller and Urey proved that you could potentially, possibly break the cycle of life begetting life. This experiment is probably responsible for lighting a fire in many a passionate biologist. However, the pipeline between the sky and the soup leaves a lot to be desired. Without further chemistry, or a mechanism to concentrate materials, the chance that the reactions that would give rise to life could happen are essentially 0. But what if we had a model where instead of the vastness of the ocean, we just had, puddles instead? This next theory is Darwin's warm little pond. Bear with me, I'm going to nerd out about Darwin before getting into that pond. To fully appreciate how crazy it was for Darwin to have this idea, I need to tell you a little bit about the man himself. Now, any viewer of my channel, especially my live streams, knows how much of a Darwin stan I am. He had writing riz. The Origin of Species is one of the best works of science communication to ever be written, and he was, compared to men of his time, relatively non-problematic. Also, his writings made him extremely relatable. What's relevant to this story, though, is the fact that even though Darwin was trained as a naturalist, looking at plants and barnacles and stuff, he managed to piece together an incredibly insightful hypothesis on how species evolve over time, a process we now know is molecular in nature. Darwin didn't know about Mendel's work in pea plants, rules of inheritance, or even what DNA was or how it functioned at the time. And yet he still managed to piece together a mostly accurate conception of evolution. He didn't address the exact origin of life itself within the Origin of Species, but had good reason to omit this. It would have been a little spicy to suggest that life didn't need a supernatural progenitor for his audience at the time, but he did toss around some ideas with his contemporaries. In a letter Darwin wrote to Joseph Dalton Hooke, he said, "But if, and oh what a big if, we could conceive in some warm little pond with all sort of ammonia and phosphoric salts, light, heat, electricity present, that a protein compound was chemically formed, ready to undergo still more complex changes." Again, I need to emphasize this. Darwin was a naturalist, fossils and tortoises and stuff. Darwin was not a chemist, not a molecular biologist. Molecular biology didn't exist back then. But his insight, even if it was just a passing thought said to a friend, showed just how terrifyingly brilliant the man was. While he never had any formal professional experience as a chemist, that much is obvious considering he didn't include the word carbon in his list of prerequisites for life. Darwin was known to mess around with chemistry with his brother in his father's garden as a kid. He loved a particular reaction so much that his classmates used to call him gas. Although his offhand remark on the idea of life originating from a warm little pond seems a little underbaked, choosing a warm little pond as his hypothesized place for life to originate in is a more compelling choice than I think even he would have given credit for. So let's take a look at Darwin's warm little pond and see how it stacks up with our checklist. Carbon and nitrogen are both checked off for similar reasons as the primordial soup, but aided by the fact that we are no longer relying on accumulation of carbon and nitrogen in oceans, but now in small ponds. So let's start with phosphorus. Phosphorus is a really, really big problem. Again, the final boss here. Remember that in oceans, phosphate contact with calcium is going to pull phosphorus out of solution as a calcium salt. In some ways, pond chemistry might make this problem worse because of evaporation. Imagine you're introducing two cats to each other. The smaller the room is, the more likely it is for the two cats to engage. Evaporation forces the room to shrink, making it much more likely for free phosphate to interact with calcium and precipitate out as a salt. However, this wouldn't be as much of a problem if there are pockets of land that can receive falling rainwater without a direct connection to the ocean. Freshwater is light in dissolved calcium, so freshwater ponds might be decent candidates for the origin of life. Another group has proposed carbonate-rich lakes as being another body of water that could get around the calcium problem. Carbonate compounds love to pull calcium out as calcium carbonate, preventing calcium from grabbing phosphate out of solution. A lot of research into ponds as an origin of life goes into trying to find exactly what type of pond would be best suited for both the conditions of the early Earth and for chemical synthesis in a pre-enzyme world. Amphiphiles. We've already seen that amphiphiles can just rain from the sky and fall into these little ponds. But ponds are an improvement to the ocean as evaporation is a super powerful way to effectively increase the concentration of your starting material. Effectively increasing the concentration of your reactants by removing the volume is going to make a whole bunch of chemistry so much easier than it would be if you had to find your reaction partner in the literal ocean. This makes the formation of cells easier, but something that I still couldn't find is a plausible route by which fatty acids could elongate to the appropriate length. Information molecule synthesis. This aspect of the warm little pond is actually what I find to be the most compelling because the primordial soup model didn't really have a way to stitch together strings of RNA. Just like with lipid synthesis, the evaporation of these little ponds does something neat to these early chemical reactors. It increases the concentration of its contents. This evaporation can force low-concentration nucleobases, all of which have plausible reasons to be inside the pond, as we've seen in the prebiotic soup model, to undergo phosphodiester bond formation, meaning you have a mechanism by which molecules of information like RNA can be made. Re-wetting the sample brings everything back up into solution, refreshing the state that chemistry can occur in, and evaporation forces more of that chemistry to happen. Wet-dry cycling in artificial freshwater ponds with purified starting material has been demonstrated to generate RNA chains up to 53 nucleotides long, which is absolutely nuts. Once you do end up with RNA, it's only a matter of time before there's some RNA strand out there that gains the ability for catalysis, the ability to do some useful chemical function, and the rest of prebiotic synthesis might just follow from there. Part of the argument for the whole RNA world thing I'll get into later. We've already seen the power of RNA in my video on pathogens smaller than viruses. There are enzymes made completely of RNA, called ribozymes, that some RNA viruses use that are shorter than the 53 nucleotides researchers were able to synthesize. So, if this model seems to check a lot of the boxes for the origin of life, why hasn't it simply consumed the other hypotheses to become the origin of life? There's a bit of an issue here. While on paper, warm little ponds seem like great places for life to have sprung from, that is assuming something very important. Did ponds exist at all in the early Earth?
Volcanoes are a thing. Volcanoes are a thing.
Guys, look, a rock! So, were there ponds that were stable enough to survive the harsh conditions of the early Earth? I honestly don't know. There are arguments on both sides, and I'm not super comfortable with suggesting I know how strong those arguments are. And, as you might be able to tell, feelings run pretty strong in the abiogenesis world. Let me read you an excerpt from this aggressive review on the plausibility of the pond hypothesis. We will call a variety of witnesses to speak against this origin of life pretender and further caution the little pondus of the statistical understanding of thermodynamics. The second law included, known since Boltzmann revealed it to us all back in the 1870s, is fundamental to all dissipative structures in the universe, and that ignorance of this law is no excuse for endlessly propounding origin scenarios that flatly violate it. We then expose this false requiem being played for the submarine alkaline vent theory for life's emergence for what it is, before playing our own overture to emergent life. So dramatic, I love it, saying little pondist like it's some kind of slur. And if these guys are warm little pondists, does that make this author a ventist? And this isn't coming from an old-timey scientist, this was published in 2021. You can kind of assume that every scientist working in this field is throwing some amount of shade at every other scientist here. Something less controversial than debating on the presence of ponds is the unique vulnerability for ponds to be bombarded with UV light from the sun. While UV light can catalyze all sorts of useful chemical reactions, as we've already seen, having a surface pond getting absolutely blasted can make the long-term stability of these useful biomolecules kind of questionable. Oxygen, and therefore ozone, only made its way into the atmosphere after life had already begun, so there would have been very little to protect the fragile molecules of life from solar bombardment. I found a paper that suggests that the accumulation of molecules within bodies of water could act as a shielding mechanism over time. So while the UV photo damage might be a problem for the first instances of biologic molecules, maybe with enough time that becomes less of an issue. Anyway, what was that about event theory for life? This next one's a fan favorite. Hydrothermal vents are fissures underwater near areas of volcanic activity. The interface between the cold ocean and the red-hot, pulsing blood of the Earth causes incredibly hot and chemically reactive veins to form. In many ways, hydrothermal vents are kind of a foil for warm little ponds. While warm little ponds are neat, tidy, and peaceful, hydrothermal vents are hostile, chaotic, and bursting with energy. There are a couple of types of hydrothermal vents: black smokers, white smokers, and alkaline vents. Black smokers are the most extreme of the bunch, originating from seawater playing chicken with magma chambers. These types of vents get hot, really hot, in excess of 400 degrees Celsius. And while I wouldn't blame you if you were skeptical as to whether anything could grow in such extreme conditions, consider the existence of thermophilic archaea that need these conditions to survive. Superheated water carries with it a ton of useful elements stripped from the solids at the bottom of the ocean. Iron-sulfur world theory posits that dissolved iron and sulfur in this unique chemical reactor could have started reactions that could build useful organics. However, black smokers just get so hot. There are some organics that fall apart when teased a little too hard. There's no way our modern organics would survive the heat of a black smoker. So while black smokers can certainly contribute to the abiogenesis question, I'm a little wary of claiming that this is the place. White smokers are a bit more mild than black smokers, and for some reason they don't really appear all that much in my review of the literature. If I had to guess, maybe the type of chemistry that happens at a white smoker doesn't scream abiogenic potential. This handy review from Nature I'm consulting for this segment doesn't even mention them, so moving on. The hydrothermal vent that I will focus on for this video is the alkaline vent. These vents are super duper mild. Instead of the violent superheating of the black smoker, we get a cozy 60 to 80 Celsius instead. Its mild heat comes from a lack of near contact with magma, instead drawing from the chemical reaction of ocean water with exposed mantle. This reaction is called serpentization and is an exthermic heat-releasing reaction that also strips water of its hydrogens and releases it as hydrogen gas, leaving behind a hydroxide ion which dramatically raises the pH. Hence, alkaline vent. Over time, dissolved calcium reacts with this liquid and forms these porous chimney structures. Dissolved minerals, mild temperature, a pH difference with the surrounding seawater, and a porous surface are all going to be reasons why these vents make for ideal reaction vessels, which I will explain in just a moment. But first, the checklist. Bioavailable carbon comes in a bit of a different flavor here, now as a metric ton of methane spewing out of this chemical reactor in addition to dissolved carbon dioxide. Nitrogen presents a bit of a problem for the hydrothermal vent. In the previous models, we needed to harness some of the most wicked sources of energy to pry diatomic nitrogen apart, just so that we could generate some useful products. At the bottom of the ocean, this chemistry is just not likely to happen. And for a long time, nitrogen would be a complete roadblock for the ventists. But there was a tantalizing finding. Within these hydrothermal vents is the single amino acid tryptophan. This is important because if you had a slew of other amino acids, you could write off the presence of usable nitrogen to a biotic source. Maybe some microbe died and coated these vents with their guts. But if you only find tryptophan, the chance that you have some actual specific chemistry goes up the roof. What's even spicier about this finding is that tryptophan isn't the only organic found in this vent. There are tryptophan precursors as well. The fact that researchers found indole suggests a specific pathway called Friedel-Crafts, which I'm not going to pretend to understand. Later research would strengthen this hypothesis by finding saponite, a key catalyst that can make ammonium easier to use. Phosphorus, oh phosphorus, you miserable element, you. While vents provide an ideal chemical reactor, the environment of the ocean itself kills bioavailable phosphorus. Ocean water is really high in calcium, which when encountering phosphorus will precipitate out as calcium phosphate and become bio-unavailable. Once again, phosphorus has ruined the fun. Or has it? Thinking outside the box, researchers have suggested that phosphate may have been bioavailable through a different mineral source, as phosphite instead. We know that there are microorganisms that have the capability to obtain phosphorus through phosphite instead of phosphates, and we do know that hypothetically, serpentization in the alkaline vent could give rise to phosphite as a product. What I'm having a lot of trouble finding is direct evidence that alkaline vents can make phosphite, as nowadays in our post-oxygen world, phosphates are far more common and bioavailable. Amphiphiles and lipids. This entire time, the vent theory has needed some clever workarounds for these life prerequisites, and amphiphiles are not an exception to this rule. In an alkaline vent environment, the conditions for chemistry heavily disfavor the formation of cells. While hydrothermal vents do have the chemistry to make long-chain fatty acids, the harsh, salty environment of the ocean is going to make it very difficult for these protomembranes to form. This does mean that freshwater ponds might have a leg up with regards to plausible protocell formation. However, the alkaline vent enables us to sequence break and potentially push the lipid problem all the way down the priority list. Cellularity is a huge accelerant for the evolution of life, as having little packets of biologics concentrates molecules and makes chemical reactions much more likely to happen. Alkaline vents have a workaround, though. Alkaline vents are porous, they have little holes in them. These sponge-like structures provide potential compartmentalization mechanisms before the prebiotic assembly of fatty molecules. Maybe vents got us most of the way with regards to the insides of cells, and the lipids themselves came from somewhere else. The surface area of these vents also provides other catalytic benefits. Certain areas of certain vents are coated with iron sulfides, which are catalytically important and can really help the conversion and capture of this bubbling chemical vat into productive organics. What makes the hydrothermal vent extra compelling is that there is a natural gradient of protons that is formed by the architecture of the hydrothermal vent. And if the term proton gradient triggered your neurons, I am proud of you, and you've been paying attention in biochemistry. Proton gradients are also responsible for driving the production of energy in your mitochondria, the powerhouse of the cell. Your mitochondria use the enzyme ATP synthase to create adenosine triphosphate, the molecule of cellular energy. ATP is really useful because it is a high-energy molecule that can donate its intrinsic energy to help power chemical reactions that would otherwise be unfavorable. The problem with the prebiotic world is that there is no ATP synthase, so no easy way to generate ATP. ATP synthase itself is powered by taking advantage of a proton pump. You can kind of think of a proton pump as being a kind of ripcord. The flow of protons itself can provide a massive amount of energy to catalyze reactions. And in a pre-enzyme world, a naturally occurring proton pump may be the way biomolecules got made before enzymes existed. And because these vents are located so deep within the ocean, you no longer have to worry about photochemistry shredding the molecules you do end up making. With all of this down, it would seem like the vent theory has no weaknesses. Hypothetically, vents make the world's first complex metabolic networks, and the rest is history. There is one weakness, though, and depending on who you ask, it's kind of a major one. Hydrothermal vents get very hot. As many of you know by now, if you're working with RNA and an undergraduate sneezes in the adjacent room, your RNA sample degrades. That's a joke I like to tell to express just how unstable RNA, a key molecule that can store information, can be. You don't even have to go all the way up to the temperature of black smokers. Even the more moderate alkaline vents can be pretty hostile to RNA formation, and you now know why there are many people hesitant to adopt a vent-based origin of life. However, once again, hydrothermal vents have a little trick up their sleeves. Again, we have the very important role of surface chemistry coming in clutch. Free RNA might be shredded by the heat, but binding to the surface of these vents can stabilize them, hopefully long enough for some productive chemistry to start happening. I really struggled to find any vent experiments that could rival the length of the RNA strands ponds are capable of producing, though. Now, if you're a chemist, you might be upset that I essentially glossed over some of the coolest chemistry to naturally happen on planet Earth, and you'd be right. The Octopus Lady has already done a video on the deep-sea hydrothermal vent and abiogenesis, exploring the chemistry of the early ocean with ChemThug, and for a deep dive on the hydrothermal vent, I could not do a better job than what they did. Go check that video out, it's awesome, and it's also a video that I'm sure would enrage the warm little pondists. Ponds and vents, from what I'm seeing, are two competing models for prebiotic chemistry. A lot of their disagreements come down to the specific particulars of the early Earth environment, as well as the ever-raging tension between tightly controlled robust experiments and the validity of those experiments. It's a never-ending, passionate disagreement between these two camps, but I think something these rivals can agree on is a mutual revulsion and disgust towards panspermia. Panspermia is the idea that life on Earth was seeded by something from space. Cosmic dust, a meteorite, something of that nature. And the reason why this hypothesis sucks is that this isn't really an explanation for the origin of life. Sure, it might explain how life on Earth started, but all this does is push back the real answer to the origin of life somewhere out into the vast unknown of space. Trust me, even if you like the idea of panspermia, it is without a doubt the least interesting explanation for how life on planet Earth originated. Think about it. Is it cooler to believe that life happened elsewhere first and we're just kind of DLC for that life? Or is it cooler to believe that through the combination of the chaotic force of chemistry and the deep time inevitability of stochastism that life emerged from the hellscape that was the early Hadean Earth? Not only does panspermia suck for aesthetic reasons, it's also kind of a bad scientific hypothesis. In order for panspermia to be compelling, there would have to be some evidence for life having been seeded on Earth, like a microbial fossil or something. To date, there don't appear to be any instances of microbial fossils found in meteorites, none that are clearly microbial and not mineral in nature, at least. And while panspermia might be viewed as a fringe, non-serious contender for abiogenesis, that doesn't mean space rocks played no role in life on Earth. Pseudopanspermia is the idea that while meteorites did not seed life on planet Earth, these meteorite collisions did provide Earth with a lot of fertile conditions. Pretty much all models of abiogenesis benefit from the finding that meteorites that crash into Earth carry with them a lot of biotic molecules preformed, as meteorites have been found to contain ammonium and ammonia salts, partially solving the bioavailable nitrogen problem. Long hydrocarbon chains that form the building blocks for lipids were also found in meteorites. And this is a last-minute addition to the script, as it was recently discovered that the asteroid Bennu contains 14 out of the 22 amino acids proteins are made out of, as well as all 5 nucleobases that build up RNA and DNA. However, the amino acids found in Bennu were found to be racemic, meaning we have a chirality problem that I'm not ready to address for like another 20 minutes. And more importantly, meteorites have an answer for the phosphorus bottleneck, the plague of pretty much all abiogenesis models. Certain space rocks contain appreciable amounts of schreibersite, a phosphorus mineral that readily reacts with water to form phosphite and pyrophosphate, usable forms of phosphorus and precursors to ATP, the cell's molecule of energy. And if you're a warm little pondist, if there's any appreciable land anywhere in the Hadean Earth, a meteorite collision with said land could form a crater that forms, you guessed it, a pond. Panspermia might be looked down upon, but pseudopanspermia ends up solving a lot of the theoretical issues with our existing models. Kind of interesting how that all shakes out. From what I can tell, ponds and vents are the two main camps to be in right now. But there are a whole bunch of alternate hypotheses out there that don't fit neatly into either camp. I'll just talk about one of these non-mainstream models. Cold start, ice world. Despite the hellscape that is the early Hadean Earth, there's some evidence to show that the early sun was quite a bit weaker than our modern sun, which hypothetically might have allowed for patches of ice to exist. This one's kind of a funky one because Ice World takes what we conventionally know about chemistry and turns it on its head. Ponds and vents have sources of energy that can be used to speed up the rate of chemical reactions. Faster moving molecules means that they can find their reaction partner much faster, making rates of reaction quite quick. However, as we've seen, this energy input has a downside, especially if your products are fragile. Ice World takes the opposite approach to vents. It's the slow and steady model that suggests that life started in very cold water. This has its benefits. A cold temperature stabilizes molecules, making them more likely to hang around for longer. Freeze-thaw cycles provide a very similar effect to the wet-dry cycles that the pond is like. Ice itself can also act like a catalytic surface, much like clay in pond models or iron sulfide in vents. Overall, Ice World is kind of a cute model, and it's definitely growing on me. So ponds, vents, ice, or something else. We've heard some arguments for these camps, but I'm sure you're wondering which I think is the correct answer here. But first, aren't we just playing word games here? At the top I said, technically the start of life was the Big Bang. Ventists want to say that life started underwater because the chemical reactions that were occurring down there were essential for life. Pondists might argue that the condition to form the world's first protocell could not be found under that much heat and pressure and salt. So, if you define the start of life as the first chemical reactions that mattered for life, you might lean vent. If you define the start of life as a place where the first protocell could happen, you might lean pond. 2, this is just a vibe I'm getting from reading all of these freaking papers over time, but I think maybe there is a middle ground where vents and ponds both contributed in their own unique way to the origin of life, an idea I'll explore near the end of this video. And finally, according to the Merriam-Webster Dictionary, the term epistemology is the study or a theory of the nature and grounds of knowledge, especially with reference to its limits and validity. And my goodness, do we have an epistemological nightmare on our hands. Not only of the enormously daunting task of trying to figure out exactly what happened 4 billion years ago, but also an epistemological barrier for me, Phi the Neutrophil. I think a lot of the subjects I've covered thus far have been pretty much in my comfort zone. I choose easy targets where scientific consensus is strong enough to give me the benefit of the doubt. However, the debate between vents and ponds is still actively raging to this day, and it's hard to have an actual opinion on this subject. In scientific matters where the facts are not settled, it can be kind of daunting to know who to trust. But here's the neat thing about reality. Absolute certainty doesn't exist for you and me. The science is the result of resolving tensions between ideas that percolate, metamorphosize, and evolve for decades. Because every single researcher actively trying to push the boundaries of human knowledge comes loaded in with their own sets of biases and intuitions. Scientific scuffles are not usually settled overnight, but over decades of publishing, conversation, controversy, and perhaps a bit of spirited name-calling. Meaning, the science is not objective fact, but a tentative truce between warring factions. Absolute certainty is an illusion we tell ourselves to quell the anxiety of an uncertain future. But this isn't a reason to fall into what I'm calling epistemic nihilism. I don't know if there's a better term for this, but when people find out that experts can be wrong, it tends to hurt their trust in scientific authorities as a whole. Now, scientists know that scientists can be wrong. Scientists, as individuals, are almost by definition more wrong than they are right. But the important thing to remember is that the ability for science to make accurate predictions improves over time, and that improvement is basically permanent and global. So, how do you worm your way out of epistemic nihilism? I think instead of viewing reality as a series of truths and falsehoods, you've got to see it as a series of probabilities instead. By acknowledging the fragile state of truth, when predictions go wrong, it might just soften the shock just a little bit. What happens when you feel like the probabilities are 50-50? This is the case that I am experiencing now. I have read the arguments from both sides, and with my limited capacity to judge them both critically, here's what I will say. The absolute subjective experience of me doing the research, using whatever articles Google Scholar wants to show me, is pulling me towards the vent hypothesis. While NASA does talk about both vent and pond theories, it does kind of feel like NASA is feeling the vent stuff a little more. Is this a product of real scientific momentum, or am I being biased in some way through the literature that I happen to be exposed to? I don't know. Gun to my head, I'd say vent. That's just what my gut is telling me. However, I like the pond hypothesis, and not just because of my man crush on Darwin. I can't get over the fact that with some bare bones starting material, you can get polymerization of more than 50 nucleotides worth of RNA in fresh water using dry wet cycles. I might not be prepared to defend how plausible it was for freshwater ponds to exist, but I just kind of like the idea. Which brings me to my belief is a composite. soapbox. As scientists, we are trained with maximum care and have belief comprised mostly of the evidence, but reality is uncertain, and that uncertainty gap is often filled in with things like affinity for certain epistemic frameworks, or, and this is controversial, aesthetics. I have an aesthetic preference for clean reactors that produce appreciably long strands of RNA. My training has exposed me to a lot of cool chemistry that RNA can do without the help of proteins. To me, the line between non-life and life is hypothetically the cleanest through the pond, and a bit messier through the vent. I am rooting for the ponds because I prefer their story. And, on the other side, there may be a non-0% of Adventist belief that life originated deep in the ocean that could be attributed to the fact that, on the face of it, vents are a lot cooler than ponds. Aesthetics might be a touchy subject, especially for people who are inclined to believe that they have an objective view of reality, but I think aesthetic preference is not something to shy away from, but to embrace. Having schools of thought and preferences motivates specific lines of inquiry. Sometimes these lead to intuitions, and intuitions might be the only thing you have attempting to navigate the dark unknown. This was all just a theoretical soapbox surrounding intellectual humility, though. Ponds and vents are not actually mutually exclusive anymore, according to some schools of thought, and we'll see how they could come together in like 15 minutes' time. We've basically covered what I consider to be the main contenders for where life originated. Now we have to shift our perspective to discussing which modern biologically relevant aspects came before the rest. This conversation is going to look a lot like the conversation we just had about location, except we're going to focus more on the chicken and egg problem of which came first. Molecules of information, or metabolism, or compartmentalization? In many models, you can't have working proteins without genetically encoded molecules, but you can't have genetically encoded molecules without proteins to synthesize them. It's complicated, we'll get into it. But before we do, it's the egg. The egg came before the chicken. This is a known biological fact. Eggs evolved during the Carboniferous period 300 million years ago, while chickens came on the scene about 5 million years ago. That particular question, as it is worded, is not a deep philosophical question. Okay, now we'll get into it. In order for life to happen, you need at least 3 major aspects. 1, a replicating, heritable molecule of information. 2, biochemistry, aka metabolism. And 3, a compartment to separate and concentrate those other aspects. Where people disagree is which aspect came first and what type of molecule embodied these aspects the most accurately. It's a little hard to talk about in the abstract, so let's take a look at what is possibly the most popular molecule with regards to abiogenesis, RNA. We've talked about RNA in passing, so let's actually define it here. RNA is ribose nucleic acid, comprised of 3 parts: a nucleobase, a ribose sugar, as opposed to deoxyribose in DNA, and a phosphate group. As we've seen in the previous section, all of these components have plausible chemistries in the prebiotic world. RNA, much like its more stable cousin DNA, is a string of 4 different bases: adenine, cytosine, guanine, and uracil. Patterns of these different bases can be seen as a kind of code. We know now that what we ended up with is a triplet code, where each combination of 3 adjacent letters can code for 1 amino acid. RNA is extraordinary and lies at the very center of molecular biology itself. In theory, RNA is the perfect candidate for the first biologic molecule. RNA can not only store information, but it can interface with DNA, providing a hypothetical stepping stone to develop the chemistry required to make DNA. It can also catalyze chemical reactions all on its own, mediating the chemical reactions that give rise to ester and glycosidic bonds. But perhaps the most striking reason why RNA serves as the ideal first biological molecule is the fact that RNA can catalyze the formation of peptide bonds, stitching together the amino acids we know were present in the early Earth to form proteins. And this isn't just a theoretical lab thing. You don't have to look beyond your own cells for the proof. Remember, the organelle, the ribosome, exists. Our modern-day ribosome relies on RNA as the catalytic core of the machine that builds proteins. And once you've solved the origin of proteins, the rest of life will surely follow. It's not hard to believe a scenario in which RNAs gave rise to proteins. Except, we have a problem. The ribosome is made of more than just RNA. It's also made of protein. Here's that chicken-egg problem. You need protein to build protein. How could the ribosome have been built in the first place without a ribosome to build the protein parts? And here's the mind-bending thing about RNA. While you need protein to be able to build a ribosome to do protein synthesis efficiently, you don't actually need the protein bit if you're okay with a system that just kinda sucks. Researchers have shown that an RNA molecule just 197 bases in length was sufficient to be able to do some very basic stitching together of amino acids, the building blocks of proteins. Hand this molecule off to the evolution tinkering lab, and it's not hard to believe that molecular complexity could radiate from this starting point. C's get degrees after all. The refinement can come later. RNA's catalytic ability extends beyond protein synthesis, too. RNAs can catalyze their own replication. While whatever exact sequence of RNA's life originally started off with is unknown, in the lab it has been shown that ribozymes completely made of RNAs can actually replicate RNAs with the use of templates. And not just tiny fragments either. One group of researchers found that their ribozyme could copy fully functional strands of other ribozymes. This self-catalyzing replication is the core of the evolutionary machine, as once you have self-replication, you have the ability for selection to really refine sequences over time. In terms of enabling all of life to happen, it's really no wonder why people want the beginning of the story of life to be RNA. We've already seen that freshwater ponds have the capability to generate RNA molecules of at least 50 base pairs long. The idea that over millions of years, under very special circumstances, a functional RNA that catalyzed a very simple self-sustaining reaction isn't a crazy one. But it's not without its challenges. And its challenges are numerous. While warm little ponds have proven that in a controlled environment, nucleotides like RNA can polymerize to very impressive lengths, in the real world, RNA is unstable. That and there are plenty of unanswered questions as to whether or not RNA that polymerizes under pond conditions could ever be usable and not just a pile of crap. This is an issue broadly with abiogenesis. There aren't many convincing ways RNAs can be made with the correct chirality. Chirality refers to the relative handedness of molecules. Amino acids, the building blocks of protein, evolved to be left-handed, whereas RNA and DNA evolved to be right-handed. The chirality of the products of a chemical reaction are really important. It's why chemists obsess over making sure that their products are enantiomerically pure. Chemicals with a wrong-handedness are not often useful, or are actively detrimental to the purpose of the product. And taking a look at the chaos of the early Earth, there doesn't seem to be an obvious mechanism by which this chirality symmetry can be broken. That, and when you start a chemical reaction with an impure racemic mix, you will often find that your products are basically unusable sludge. That hasn't really stopped scientists from thinking about this problem, though. There are plenty of hypotheses that I'm not equipped to evaluate, but I wanted to mention for your consideration. The first hypothesis I wanted to mention was the hypothesis that I myself believed up until doing research for this video. Pseudopanspermia comes in clutch once again, as meteorites that hit Earth tend to come loaded in with L amino acids, meaning that, hypothetically, L amino acids could bias the formation of dRNA. Seems logical to me, except for the fact that the ratio of L to D amino acids in meteorites is much closer to even than I initially thought. Bennu, which I mentioned before, is proof that not all meteorites have a chiral bias. Bennu delivered a mixed racemic mix of amino acids. There's also some debate that I'm not qualified to evaluate about whether RNA biased amino acids or whether amino acids biased RNA. My next thought was maybe this was all just a coin flip. If, in the pond of life, dRNA got a head start, maybe the formation of dRNA would further encourage the formation of dRNA and feedback from there. This is also kind of a stretch for the whole sludge thing. Another thought is that maybe life didn't need to evolve a preference right away. There's a paper that found that lRNA ribozymes could polymerize dRNA, meaning both forms of RNA could have been useful in early life. This is so cool. But much like how panspermia pushes prebiotic chemistry into space, this hypothesis doesn't really answer the symmetry breaking problem. And finally, maybe the symmetry breaking event happened way, way before life. All the way back to, you guessed it, the Big Bang. For reasons I cannot explain, it is hypothesized that the physics of the universe itself is left-handed, and that symmetry break could have biased biological chirality as well. I'll just read a little snippet of this handy review here. In elementary particle physics, parity violation during beta decay of nuclei may have led to a slight increase in the ratio of L to D enantiomers. It is also suggested that polarized synchrotron radiation from neutron stars may have influenced the proportions of these two enantiomers. Chirality in biology is a really interesting problem, and if you want to hear my thoughts on the implications of so-called mirror life, the hypothetical organisms that evolved opposite-handed chirality to life as we know it, go ahead and subscribe. I'll definitely get to it, eventually. And membership if you want perks like livestream emojis and members-exclusive non-science content. And if you want to support me on Patreon, that's now a thing too. Other models of abiogenesis ditch the idea of the first relevant biomolecule being something we're familiar with in modern biology. I'll talk about just one model, one in which a hybrid RNA peptide preceded RNA. Peptide nucleic acids have nucleobases, which are parts of RNA that allow for storage of information. But instead of the ribose sugar phosphate backbone, they're instead linked by repeating units of the amino acid glycine. PNAs are more stable than RNA. They exhibit good base pairing, they don't suffer from the same chirality problem as RNAs do, so why aren't we all on the PNA train? Since PNAs don't really exist naturally anymore, it's kind of hard to know how this molecule could have ever been a stepping stone into the RNA world. PNA world is a bit of a weird one, and not something that a lot of people put heavy investment in, for now at least. While RNA and RNA-type molecules are the premier molecules for information in a prebiotic world, I do want to give a brief shout-out to the amyloid-first hypothesis, because it wouldn't be a Find the Nutrifil video if I couldn't talk about prions. Prions are special proteins that, when folded in a certain way, causes other similar proteins to misfold. These misfolded proteins aggregate in amyloid sheets and are super stable and resistant to degradation. They build up in neurons, leading to their death. Prions are responsible for some of our scariest neurodegenerative diseases, and while these misfolded proteins are largely incompatible with life as we experience it, their ability to resist harsh chemical conditions and propagate information by misfolding proteins makes them an intriguing option for a pre-RNA replicator molecule. Not only that, but amyloids also tend to be homochiral, as in order to get that really stable structure, you need all of the aggregating molecules to be the same chirality for optimal packing. But big whoop, even if you generated these amyloids, wouldn't they just be a bunch of useless junk? Certain amyloids have actually been shown to be catalytically active, meaning just like RNA and conventional proteins, amyloids can also do a bit of chemistry. So you can maybe imagine a situation where short peptides generated by the prebiotic Earth formed superstable amyloid structures that provided RNA with a homochiral foundation to bind to and potentially be protected from the harsh environment. Amyloid first is a very niche and speculative hypothesis, but I mean, come on, that's awesome. There are glaring issues with a replicator-first worldview, and it's one that RNA World opponents can't overlook. Opponents of this angle seem to favor the metabolism-first model, which states that before information molecules had to have been a bustling environment of chemical synthesis, metabolism. This is the model that most cleanly transitions from the hydrothermal vent theory. These vents produce a local environment where all sorts of insane chemistry is allowed to happen. Maybe some of these resulting products are short proteins that help RNAs synthesize, stabilize, and evolve. Once RNA and crude peptides evolve to take over some of these metabolic functions that vents can provide, life can escape. We've already mentioned iron-sulfur surfaces and the porous microstructures within these vents provide areas in which, hypothetically, RNA could form. Although again, experimentally, these polymers have been quite short. And while information and metabolism first gangs are fighting, over here we have the friendly lipid world theory, which postulates that lipids were first on the scene. Lipids, or fatty membranes, pretty much define cellular life. Remember how having an ocean dilute all of your stuff is really bad for productive chemistry? Lipids provide a way to enclose and concentrate chemicals to the point where you can sort of force their reactions to happen, or at least heavily encourage them. While lipids aren't exactly the most exciting molecules catalysis-wise, they do offer a supportive boost to either information first or metabolism first hypotheses. The other interesting properties of lipids to note is that lipids are everybody's unproblematic faves. If you just have lipids in water, they will form cells. There's not a whole bunch of scary contingencies that need to exist, like for RNA. Except again, the Ventists believe that the porous microstructure within the vents acted as a compartment before lipid-based cells were ever a thing. Meaning to them, life, going back far enough, may have started within rocks. Still others believe that before lipid compartmentalization came coacervates, or liquid droplets. Instead of compartments being separated by an oily membrane, these cells would be separated by a combination of peptides and RNA. This isn't a totally crazy idea, as protein-based compartments are still a thing in modern biology. Take the rabies negri body, a viral factory that separates liquid from liquid but is entirely made of protein. You don't have to look all that far from your own biology either, as your own nucleolus is also a liquid coacervate. So, theoretically speaking, liquid coacervates may have preceded lipids as the initial structure for organizing biology. Which came first, I think, is a question that is useful to push the boundaries of what we know about the abiogenesis problem. But where I think research is heading towards is evolving, mixing, and matching all of these different stances. I remember RNA World being the predominant hypothesis when I was learning biology as a young cell. Viewing the abiogenesis landscape evolve after all those years makes it more clear that alternate hypotheses, including mixed models, have entered the conversation. You can imagine a situation where perhaps vent chemistry provided the raw material. Maybe a lot of chemically complex material from the vents floated to the top of the early oceans. Maybe some of these molecules found themselves in a land crater, and or near a volcano where that soup could escape the enormous ocean. Then, through UV photochemistry and wet-dry cycling, polymerization of useful biologics could occur on clay surfaces. That is one possible narrative that could exist, as you take the best of all worlds. The powerful chemical reactor at the bottom of the ocean, with the bioavailability of molecules locked inside comets and volcanoes, the wet-dry cycling of the little pond, to stitch together more complex strings of RNA or RNA-like molecules. Again, the best of all worlds. Except panspermia, because I think that sucks. Of course, there's a lot more to talk about when discussing the order of these milestones, but I need to move on. Every time I do explicitly state in a video that I need to move on, I do end up getting a lot of very supportive comments telling me they'd watch me no matter the length of the video. And those are very nice sentiments to have. But if you've ever watched me stream, I've made one of my deep and primal fears known to my audience. This is not a joke, okay, real talk for a second. I have a real, actual deep anxiety that I will die before I cover everything I want to talk about in biology. And the more time I spend on one video, the closer the stem cell stock from which I differentiated from gets to becoming unviable. With all of that being said, we are finally done with the prebiotic chemistry portion of this video. Begone chemistry, and hello our beloved biology. But where is this handoff between chemistry and life? That's more of a philosophical question, but what isn't a philosophical question is the handoff between randomly generating polymers of stuff and actual, real evolution. The exact frame of history where chemistry developed a molecule with a capacity to replicate and transmit inheritance, real, actual molecular evolution can begin. And once evolution begins, progress to life will rapidly accelerate. I can hear the RNA world stands waiting with bated breath. Yes, RNA is a molecule that contains information and is heritable. But is RNA the only molecule of inheritance? And I'm not referring to DNA. I think people are pretty comfortable with the development of DNA happening much later in biologic time. DNA is more complex than RNA, harder to build, so most people just assume that RNA must have come first. No. The molecule of inheritance I'm talking about is the humble lipid. Lipids are capable of imbuing a system with properties of inheritance, and therefore a primitive version of evolution. It's just a different kind of evolution than RNA. If RNA can code for a kind of digital information in its code, lipids can code for a kind of analog information. A physical representation in the fact that when a lipid cell splits into two, each cell will inherit a portion of the paternal cell, its inside contents, and membrane contents. This is the concept of compositional inheritance. But wait a second, how would primitive cells grow and divide? Modern cells require a whole bunch of machinery like the actomyosin ring to form at the border of daughter cells. This ring of proteins will constrict and literally squeeze two cells apart. But this ring of proteins is in fact encoded by the genome, which doesn't exist yet. The neat thing about lipids is that these little fatty bubbles don't need proteins to divide. Just from their physical properties as modeled by the GARD formula, which is something, math, yes. A general kinetic model for the homeostatic growth and fission of compositional assemblies. Basically, GARD is the idea that if you put a bunch of different lipids in a vat with some stuff in it, that over time, the most stable structures and compositions will emerge as the cream of the crop. These lipid vessels can absorb lipids, leading to division when they get too big, or they can crash into each other and merge. Then the stress of growth can often lead to them splitting. Over time, these random matings of different lipids will result in some lipid cells being more stable and better at replicating. Has this lipid protocell evolution ever been observed experimentally? Yes. The cold start ice world folks got some experimental proof that freeze-thaw cycles not only led to a selection for a specific composition of lipid in a protocell, but also a selection for encompassing genetic material. Evolving lipids in freeze-thaw conditions led to surviving vesicles getting better and better at capturing DNA over time. DNA? Wait, if we're looking at lipid inheritance, why aren't we looking at RNA instead? Wouldn't that have made more sense in the context of abiogenesis? RNA World and all. I read this paper up and down and could not find an explanation as to why DNA was used instead of RNA. If I had to guess, it's because DNA is built like a Nokia brick phone and RNA is built like a Cybertruck. Their paper brings up an interesting point. Lipid evolution needs to dovetail with RNA evolution at some point. And we've already seen that in an ideological vacuum, RNA first as a model is really, really tempting, but kind of difficult to justify for reasons we've already gone through. But as I've been reading about protocell formation, something very interesting started to emerge. Not only are lipids these amazing little bubbles that could evolve the ability to evolve before RNA had a chance to, there's a chance that lipids actually helped RNAs evolve themselves. It has been shown in at least a couple of different labs that lipid encapsulation makes it easier for RNA to fold. And if RNA can fold, it can stabilize and survive. And if it folds productively, then that RNA can develop an actual function, a function that selection can really act on. This is also something that can be observed in a lab. Researchers took a ton of different catalytic ribozymes whose function is to attach amino acids to themselves and saw that when these ribozymes were encapsulated, that they tended to function better than when they didn't have a lipid membrane around them. Other researchers have found that the composition of these lipid cells could improve the activity of ribozymes whose job it is to repair breaks in the RNA backbone, which is kind of a baby version of replication. And this really ignites the imagination. Lipids can help RNA ribozyme activity. And while nobody, as far as I could tell, has proven that lipids could help RNAs replicate themselves, the proof of principle is at least there. In two separate instances, lipids were able to help RNA ribozymes do their job. I wouldn't be surprised if it were only a matter of time for someone to find a setup where lipids could help simple RNA ribozyme polymerases to do the same, polymerases being enzymes whose job it is to replicate genetic material. Lipids improve RNA ribozyme function. This is one half of a hell-raisingly powerful idea. We just learned that lipids under certain conditions can naturally evolve to get better at capturing nucleotides. DNA isn't that different from RNA from a chemical perspective, so let's just assume this is true for RNA as well. If lipids can evolve a capacity to capture RNA, and that captured RNA is capable of gaining or improving some kind of function by those lipids, if that captured RNA would return the favor to lipids, well, I'll give you a second to figure the rest out. Do you see it? Now we have a positive feedback loop, something that resembles an engine. An engine that you can plainly see could potentially power the rest of life itself. Most fail, most die. That is the reality of natural selection and evolution, but some of these protocell monkey typewriters tinker with RNAs for so long that eventually they start being able to do more stuff like replicate. Hot off the presses, researchers found a 45 nucleotide long self-copying ribozyme, which, need I remind you, is well within the size range of the RNAs researchers have generated in freshwater pond conditions. Replicating ribozymes get permission to extend, mutate, gain new functions by evolution. Some of these functions might synthesize short proteins, some of which bind to RNAs to form the actual protoribosome, which gets better at making longer proteins. And all of a sudden, this runaway engine of selection grows and becomes more and more robust as RNA gains the power to convert RNA into DNA, which, by the way, we know is possible in a lab, which is insane and crazy. All of these chemical networks emerge, giving birth to new functions and crescendoing complexity until we have an actual, real cell. Hallelujah. The beginning of it all. And, at least for me, this all comes down to one stipulation. RNA ribozymes can do it all. Self-replicate, catalyze protein production, even convert RNA to DNA, all without the help of any protein. But can RNA ribozymes catalyze lipid production? Can RNA return the favor to lipids and actually jumpstart this enormously powerful positive feedback loop? No. I couldn't find a single ribozyme that could even theoretically make a whole lipid from starting material. But ribozymes might not have had to. We've gotten quite lucky with the chemistry of lipids, as a lot of the chemistry required to build one can be found in the environment. Scientists have found that even the difficult conversion between simple lipid to our modern phospholipid can be catalyzed by natural environments like hydrothermal vents and soda lakes. An experiment with model protocells shows that molecules like RNA could help lipid protocells absorb other lipids, enabling them to grow. In theory, a replicating RNA could increase the osmotic pressure within these cells, making it more likely for these cells to incorporate more lipids to keep the total stress of the cell low, and when they get big enough, divide. And in the early days of life, maybe that was good enough. Life never had to be perfect, and with all of the swirling chaotic chemistry around the cell, maybe a gentle nudge towards growth and division was all that was needed to start this engine. But life is horrendously complex. And the problem with thinking about probability in the form of waiting for infinite monkey typewriters to type out the complete works of Shakespeare is that there aren't infinite monkeys, and they didn't type for an infinite amount of time. You might resist the idea that the complexity of life could have emerged from simple prebiotic precursors. Let me show you this problem in a different light. What do you think is the minimum number of genes a cell needs to do just the most basic functions of life, meaning information replication, growth, and division? Right now, as far as I know, research has found that the minimum number of genes a cell would need to replicate molecules of information, divide, and synthesize lipids is 42. That's it. The monkey typewriter hypothetically just needs to write out the complete works of 42 genes to be able to replicate, grow, and divide. Now, keep in mind there are a lot of caveats to this 42 number, and also this cell isn't really alive, but it can serve as a hypothetical starting point for evolution to do its thing. I can't prove to you that the monkey typewriter did it, but what I am saying is, if I had to bet, I bet the monkey typewriter could do it. So, how did life begin? If you ask me, and only me, the early Earth was a large chemical reactor that combined raw materials from the atmosphere, volcanoes, and comets. Prebiotic chemistry happened through some combination of hydrothermal vents, ice, and warm little ponds. Over time, this crude chemical reactor would give rise to primitive lipids and short RNAs that stabilized each other, enabling selection to act on the protocell as a whole. RNA ribozymes then took care of almost everything else, self-replicating, catalyzing protein, lipid, and DNA production, failing until the first true cell emerged, and the rest is history. If you couldn't tell, this is, of course, a tiny bit of simplification. There are going to be parts of this video that are going to be proven wrong. You don't need to tell me in the comments that there's some research that contradicts what I've said here. I know, and if you do leave a comment correcting me, you must call me a warm little pondist, so think twice before you say anything. I think of biology as a piece of candy, an infinitely deep and complexly layered piece of sweet candy, and every layer of biology that you manage to taste your way through exposes new and delicious flavors. Well, some of those flavors include memorizing the Wnt pathway, if you're a biologist, and I can't claim that all flavors are just as delicious and seamless as others. But you get my point. At the center of the biology candy is an answer to the origin of life, the start of the grand story that is evolution. There is a real chance that with all of humanity's efforts that we will taste the center of this incredible candy. But there's also a chance that when we voraciously bore through these layers that we will find that the center is hollow. That the entropy associated with time has destroyed the evidence of there even being an answer to the abiogenesis question. Humans took too long to invent science to ever find the answer. And for many people, this possibility that we will never be truly sure of how life originated is kind of fatal for their desire to further investigate this problem. We will never know exactly how life on Earth originated, but that doesn't mean we can't have a working model with strong plausibility that is consistent with reality as we understand it. New research, new models, new techniques have entered the conversation since RNA World was the only serious contender. We're learning more and more about the conditions of the early Earth. We're learning more and more about the types of chemistry that are theoretically possible. We're learning more and more about how minimal life can really get away with being. Molecular biology as a subject is less than 100 years old, and we've, in that time, went from not knowing what DNA was at all to being able to precisely edit life's blueprints, all within the lifetime of some people still alive on Earth. To believe in our limitless potential as humanity is a bold, optimistic outlook, and to be optimistic is to be brave, because it's easier and less painful to give up trying. It very well may be the case that we don't have the capacity to build these hypothetical models of very strong plausibility. But we don't know that for a fact, and I love the flavor of the unknown. As we make our way through the layers of vanilla, chocolate, and strawberry, the center of this candy always seems just a few layers away from where we are now. But time has shown again and again that every time we dig, we just end up finding new, more complex layers. Lychee dragon fruit, caramel popcorn, Dubai chocolate matcha latte, maybe even some really weird ones like Sichuan hot pot. I don't know what the flavor in the center of this candy is. I don't even know if the center is still there. Reasonable people may assume that the center is divine, but I prefer the anxiety, the mystery, and the challenge of the unknown. I do not know what lies at the center of biology. I don't know how many layers we have yet to peel back. But what I do know is that I will be here, enjoying every flavor until my own biology stops me from doing so. That's it. That's where the video ends, right at the beginning of this beautiful life. Oh my god, that was a long one. Oh my goodness. Thank you, especially if you have managed to reach this point in the video. Thank you especially to my Patreon and YouTube members for your patience. You guys are all rock stars.